Hello, everybody. I'm just going to introduce Professor Patrick MacPhail. Patrick is an emeritus professor, which for me says it all. It means the university has recognized him as an incredibly able researcher and knowledgeable fellow all around. He's published books, published more than 100 papers. He's done a medical PhD, which again, in my book is something incredible to do. In America, doctors qualify with an MD, which is a real doctorate in our country. But here, to get a real doctorate, not an honorary one, doctors here get an MBBCH, which is an honorary doctorate. But Patrick has a real one. He was also professor of medicine for a number of years at Wits, and you simply can't do better in academic life than Patrick has done. His PhD was on iron metabolism. He's going to tell us a little bit about one of the most interesting aspects of iron metabolism. Over to you, Patrick.
Oh, thanks very much. Haemochromatosis is something of a daunting word, and it's actually a bit of a misnomer, but I hope you'll enjoy this talk. I've tried to bring in some history and some aspects of the Viking world because it is so important to the whole thing. Haemochromatosis is something of a misnomer, and this gentleman, a German who is very precise, as you can see, very tidy man, he noticed that on doing postmortems on people with this condition, that the organs were a funny sort of blood color, and hence the name, heme for blood and chromatosis for color. And he recognized at that time that it was due to iron. However, the condition had been recognized some 2 decades earlier by some French physicians, and they described a syndrome of diabetes associated with bronze coloring of the skin. Now, this is a bit of an exaggerated example because the bronzing here is very, very bronze, but it really, as you can imagine, it varies quite considerably. And in fact, in some cases, doesn't appear at all. It's a mixture of iron and melanin in the skin that gives this picture. And for some reason, iron stimulates melanin production. The reasons are not clear. So that's why it has this rather scary Greek name. So what is it? Well, it's a disease of a systemic iron overload, and it's genetic in origin. And one of the iron genes becomes mutated and gets passed on from parents to children. And it happens mostly, almost entirely, in people of Western European origin. It usually doesn't start off with anything. The people are perfectly normal until about the age of 35, 40, or maybe even later, when they start to develop a whole wide range of disturbing symptoms, starting with things in young people, often starting with impotence as being one of the causes of impotence in a young man, diabetes, heart failure, cirrhosis of the liver, and eventually liver cancer as a result of the cirrhosis, arthritis, and strange feelings of fatigue, and so on. Well, now, why should it be of interest to people in this part of the world? Well, most of the people listening to this are of Caucasian origin, and their ancestors came from Western Europe. And genetic studies show that in South Africa, about 17% of the white people here are carriers of this gene. And at least 1 in 100 are homozygotes, which mean they actually can express the disease in its full expression. So what does the disease look like? Well, here's just a schema which tells you more or less what the things are. It starts, as I said, between the ages of 35 to 55, and it has a stepwise progression, getting worse and worse. And obviously, there's a lot of iron in the blood, and that causes organ damage. And we'll come on to why that is. First and foremost, the liver is affected, and you get a big liver with cirrhosis and carcinoma. But all these other things are also things that you can pick up from time to time. The ones that I've starred red are the ones that occur most commonly. And you'll see that they are liver, chronic fatigue, and cardiac failure. The sex hormone problem is an interesting one because it actually damages the anterior pituitary gland, and that gives rise to hypogonadism, which happens in most males and females. In females, it presents as amenorrhea, and in men, it presents as impotence. So it carries a wide range of problems, and you'll see why as we go through, why every organ is susceptible to something. This is a picture of a young man with hemochromatosis. He's youngish, and you can see that although he's jaundiced, you can see his eyes are quite yellow. So his skin is quite yellow, but in the background, there's this bronzing of the skin. And we were always taught to look at the forehead because that's the place where you're going to see it most. He also has an arthritis affecting the first 2 digits of his hand. And this is characteristic of hemochromatosis. And they call this the iron salute. And the reason is that if you make a fist and you've got very severely damaged joints between your finger and your hand, you can't flex them. And so you get this picture here. And I wonder how many of you are doing this with your hand to see whether in fact you have the iron salute. Sometimes called the iron fist, which is the name of a Marvel comic involving a rather bellicose individual. And if you look at him, I wonder whether he hasn't got hemochromatosis too, because there he's got these unusual changes in his hand, and he's rather bronzed. So perhaps he also has hemochromatosis. More about the kind of man he is later on. So how does this all happen? Too much iron? How does that happen? How can you get too much iron? People know about iron deficiency. Why suddenly too much of it? And where does it come from? And how can iron damage so many organs? And why is it so common? And is there anything we can do about it? Well, I think to begin with, you have to understand something about iron. Iron is a highly reactive, but very useful and very harmful metal. It's one of the transitional metals, and as like other transitional metals, it can exist in a situation where it is in the ferrous, Fe2+, or the ferric state, and it's able to shuttle between these two. And when that happens, when it moves from ferrous to ferric, it gives off an electron, which is an extremely useful thing in other reactions. And this is used biologically in a lot of situations. It can also be recharged by adding an electron to it, so it would move from the ferric to the ferrous. So it's like a battery in many ways. And it's useful in, to date, over 2,500 different reactions that take place in the body. So it's a very vital part. There's some people who say that iron, without iron, we would never have existed. Not quite true, because in fact, copper can do almost the same thing. And if you're a crab, this is what copper does. That's why crab's blood is blue and not red. So it's got many functions, and these are just a couple of them. Oxygen transport, of course, we all know. Hemoglobin, the red stuff in your blood that carries the oxygen. Myoglobin, the stuff in the muscle that provides the oxygen there for muscle function. Energy transfer, if you might have heard, it produces, the end result is ATP, which is what we use for our energy. It's involved in DNA synthesis, and it's also involved in the immune system in many factors. But it may be harmful, and that's the problem. And so what has happened is that what happens when it becomes harmful is it produces free radicals. Now, these are either oxygen or hydroxyl radicals called superoxide or hydroxyl radicals, which lead to lipid peroxidation and membrane damage, cell death, and DNA damage. But to keep us safe, many proteins have evolved to handle iron. And I'm just going to mention 3 of the many, many proteins that are involved. There's a carrier called transferrin, which carries iron around in the blood, moving it from one organ to another. And it's a highly organized system with an enormous capacity to carry iron and a very safe capacity. We'll see why in a minute. And then there's a protein called ferritin, which stores iron in a safe way. It's an ingenious mechanism. It's like a basket, and it packs the iron into it in a way that keeps us safe. And then a fairly newly discovered one called ferroportin, which is a transport protein that moves iron out of cells. Other ones here, like DMT1, move iron into cells, but this is the only one we know that moves iron out of cells. So if we were to look at a patient with hemochromatosis and to do some measurements in their blood, if we looked at their transferrin, we'd find that it was saturated with iron. Now, normally it's below 45% saturation, which gives you a lot of capacity to carry iron if you need to. But once you get to over 60%, it becomes, if you see a patient with over 60%, then it's very suspicious of hemochromatosis. It can go up to 100. But at about 60%, transferrin starts to fail, and you get free iron present in the bloodstream. And this is called non-transferrin bound iron. And this is dangerous. And the body has mechanisms to try and get rid of it as quickly as it possibly can. And the organ that suffers the most is the liver, because that's the liver's job, is to try and get rid of junk, alcohol included. Ferritin is also a measure of iron stores. Although it's inside the cell where it stores the iron, it also is in circulation, and we can measure that. And that's measured as serum ferritin, and it parallels the amount of ferritin there is in the body. And normally it's under about 200, but in hemochromatosis it can go up to many thousands. In the old days, we couldn't really make a diagnosis of hemochromatosis without doing a liver biopsy, but that's fallen away now. MRI has replaced it, and it's a very accurate way of determining what your total body iron is. Now, this picture may be a little daunting, but let's introduce you to those major characters. First of all, there's transferrin, which is the carrier that carries blood, as we said. Then there's ferroportin, which I've shown here as these purple tunnels, which is the exporter of iron from wherever it's being exported from. In this case, here it's being exported from the gut mucosa. That's the lining of the gut, because obviously iron comes in your diet and comes in through the gut and is transported into the bloodstream by ferroportin, where it binds to transferrin. And then there's ferritin, which exists in the cells here and here, and stores any surplus iron that may come in. So this circuit here is what we call the iron circuit. It shows iron coming in from iron absorption and going, most of it, to the bone marrow. Like 80% of it goes to the bone marrow to make red blood cells. These red blood cells, when they get old, around about 120 days, they get eaten up by the macrophages. Now, what are macrophages? These are cells that are ubiquitous throughout the body, but they're found mainly in the spleen. But you can do without a spleen because there's so many other macrophages around that they can do the job for you. And collectively, these are called the reticuloendothelial system, RES. And they pass the iron back into the circulation. They're also able to store it, obviously. And in fact, this is the amount of iron that comes by this route is about 20 times the amount that comes in from this route. So in fact, we conserve iron all the time. And interestingly, and you see on this picture, there's no way of excreting iron. The only way you can get rid of iron in big quantities is to bleed. But obviously, cells that leave your cells on the surface of your skin have iron in them, and as you lose those cells, so you lose iron. So there's a daily requirement of iron that comes in to keep this system going. Now, in order to understand the problem in hemochromatosis, we have to look at an original finding founded by my two mentors, Bothwell and Finch, who were both famous for their work in iron in the last century. And in the 1950s, they showed that the bigger your iron stores are, the more iron you absorb. And reversely, the more you need to make blood, in other words, erythropoiesis, the more you need to make red blood cells, the more you absorb iron. And for years, we searched around to try and find out what it was that controlled this system. But it was not until nearly 50 years later that hepcidin was discovered. Now, this is a funny little polypeptide that appeared in the urine, and people had been aware of it for some time, but nobody knew what it did. And so they gave it this rather strange name, hep because it came from the liver, and cidin because for some reason, and a reason still unknown why, it's able to kill bacteria. In fact, it seems as a side function to keep the urine sterile, which is interesting. But why, we have no idea. But it's secreted by the liver. And in fact, what it does is it's able to block iron release by ferroportin. And so it is, in effect, a hormone. The definition of a hormone is an active substance that's secreted in one organ and acts on another. And that is what it does. As I said, it comes from the macrophages. It blocks the macrophages where the ferroportin is. And it also blocks ferroportin at the duodenum, which is the major site of iron absorption. But it comes and it's made by the liver. So let's go back to our picture. Here we've put hepcidin in. And we see here that diferric transferrin, that is transferrin loaded with iron, is able to act on something that I've just called an iron sensor. For the meanwhile, we'll just call it that. That sensor sends signals out to a gene in the liver called hemp. And this gene is responsible for making hepcidin. And the hepcidin, if it's present, can either block release from the macrophage, or it can block release from the mucosa, so block any iron that is will be absorbed. Hemp can also be affected by other things like inflammation, hypoxia, and erythropoiesis. And this is interesting because if you go up and if you live in the Andes and you are constantly slightly hypoxic, you are inhibiting hepcidin. And so those people have a low level of hepcidin and a higher level of iron all the time. So now what happens in hemochromatosis is there's a problem here. This iron sensor is not working properly. And as a result, hepcidin is not being secreted. And as a result, iron pours out of the two places where it comes from the gut and from the reticuloendothelial system, from the spleen, the macrophages. And as it pours into this, it has to be bound by transferrin. But after a while, transferrin becomes saturated. The absorption here being about 2 to 3 times normal, the transferrin becomes saturated. And when it becomes saturated, you get free iron, so-called non-transferrin bound iron. And that free iron is dangerous. And so it needs to be mopped up. And it's mopped up mostly by the liver, but also by other organs, notably the pancreas, the heart, the pituitary, the skin. But in fact, almost all iron, all organs have the ability to trap non-transferrin bound iron, probably because it is such a dangerous substance. So this is the situation that is happening in hemochromatosis. You've got excessive amounts of iron coming into the circulation and being distributed to all sorts of organs. And of course, it takes time. That's why it doesn't give you any problem until you're 30 years. Well, let's have a look at this iron sensor and see what it is. Well, don't be alarmed by this. Every one of these little symbols is a protein involved in iron metabolism. And every one of them, if it goes wrong, something goes wrong. But this here is the one we're talking about, HFE. It's in close association with the transferrin receptor. And here we see iron on transferrin being accepted by the transferrin receptor. It then sends a signal through to this protein, which reacts with this protein, which then, through a whole series of things, goes to the gene and produces hepcidin mRNA, and eventually the hormone hepcidin, which is excreted into the circulation to do its job. Well, now this, as we see, is the one we're talking about, because this is the most common of all of them. But in fact, if any of these things go wrong, it will produce a kind of hemochromatosis. So here we have one called juvenile hemochromatosis, fortunately very rare, and it's much more devastating because it happens in people at the age of 15, 16 years old, but luckily it's a very, very rare condition. Complete inability to actually, hemp can get damaged just in the way other genes can get damaged, and this produces another kind of juvenile hemochromatosis, which looks very similar. And this transferrin receptor, the other transferrin receptor 2, can also be affected, and this can give rise to a third kind of hemochromatosis. But as I said, these are very rare, and although we've got a whole host of them here, the one we're looking at the moment, and why are we interested in it? Because it causes over 90% of the cases of hemochromatosis. We know what the gene is, and we know what the actual mutations are that cause the problem, but more about that later. The situation is you've got a very low hepcidin, very low hepcidin, and as we've said, it's associated with a high transferrin, high ferritin, and iron loading of the liver, heart, pancreas, and so on. And it causes most commonly in Caucasians, and the symptoms arise in the latter half of life. Now, this gene is recessive, and we'll talk about what that means in a minute, but in fact, you have to have 2 copies of it to have the full-blown disease. Now, this is the little protein that's called HFE that we've been talking about. It looks very like the histocompatible antigens, but we will leave that for aside at the moment. But the two important mutations that we know of is this one here, 282Y, and this causes this molecule here, called beta-2 microglobulin, is unable to fuse to the thing, and it knocks it over, it collapses, and in fact, because it's not functioning, the body reabsorbs it, so in effect, it's not expressed. But this one here works differently. This one seems to interfere with, the HFE's binding to transferrin, to the transferrin receptor. And we don't really know why, exactly where it is, but a number of mutations have been described in this region, and they all seem to have a similar effect. Phil has given you a very good idea about how all this goes, but let's just revise what happens here. Here is DNA, and here are all the base pairs, which, as you know, are complementary. Here, for example, U always mixes up with A, and T with A, so on, so on. And this is the genetic code. It gets read by it gets read by a system called transcription, which produces messenger RNA, which leaves the nucleus and goes into the cytoplasm, where it meets the ribosomes, and there it meets transfer RNA, which has the same code on it, but carries with it an amino acid. And these, in order, latch on to the code, and so the amino acids bind together to produce a growing chain of protein, which eventually gets released to form a protein. And the transfer RNA goes off to find another amino acid and come back and do the job again. An ingenious and remarkable system. Now, what's wrong in hemochromatosis? Well, it's mostly the C282Y mutation. And here we can see the code for cysteine is usually TGC, and the G here has been replaced by an A. And so instead of getting cysteine, you get tyrosine here. And these correspond to the 2 copies. So C means cysteine gets replaced with tyrosine, and it happens at the position 282. That's the 282nd amino acid along the length of that particular hormone. that particular protein. So now what happens about, what's this business all about, heterozygotes and homozygotes? Well, this is what we would call a heterozygote. Here's a chap who carries the gene, but his other pair, his other copy of it, is normal. Now, he would have got one of these from his mother and one of them from his father. And so he got a normal one, say, from his father and an abnormal one from his mother. He now has children with this lady who, in fact, is a normal person. So he's what we call a carrier, and 50% of his children will be carriers. So he's what we call a heterozygote. On the other side here we see a homozygote. This lady's got two copies. He got one from her mother and one from her father. And so she's got two copies of this gene, and she's therefore has the disease, and hence the red head that she's got. She marries someone who's normal, and they will produce children. Oh, sorry about that. They will produce children who are all carriers of the gene. Now, we did mention this other one here called HD. And interestingly enough, if you combine these two together, as you see here, that's called a compound heterozygote. And in fact, they have hemochromatosis, but not to quite the same extent that this person has hemochromatosis. The disease seems to take a little longer to get going, and it seems to be less severe, although it can be fatal. So it can be a serious condition. So it's not nothing. Now, how often do we see these things? Well, in Ireland, 10% of the population is heterozygous. That means that about over 1 in 100 of Irish people actually have hemochromatosis. They have the condition. In the French, it's a little bit worse. In South Africa, about 17% of the white populations that we've looked at are heterozygous, and about 1% have two copies of the gene and therefore would be homozygous. People of mixed race in South Africa, not surprisingly, have lower things. And if we look at Europeans in general, this is about the kind of figure if you include all people from Europe. The H63D mutation, however, is a different story. It's much, much more common. And it's not only common in Europeans, it's common in all people in the world. And so to be homozygous for this condition is much more common, although the disease that you get is much, much less severe. So where does this thing come from? Where does 282Y come from? Well, right at the beginning, they said this wasn't a genetic disease at all. This is just too many Irish people having too much alcohol. It's all due to alcohol. But that in 1970s, a French worker, Marcel Simon, found that there was a tremendous amount of hemochromatosis in Brittany. And he thought, being a strong Celt himself, he thought this must be a Celtic disease. Now, the Celts were a small tribe north of Switzerland in about BC 2000, and they spread throughout Europe. And he thought that there were pure remnants of this Celtic population in Ireland, Scotland, Wales, Cornwall, and in Brittany, where hemochromatosis is particularly common. But other information didn't stack up. A kind of analysis called haplotype examiners seemed to point out that it was unlikely to be a Celtic origin, and that the origin of this disease is much more recent, about between 5 and 700 years after the birth of Christ. This is what they thought happened, and in fact, this is what did happen, is that this tribe that started off down here then spread throughout Europe, and of course went into Britain and went the other way as well, although this was a much later migration. And they thought that this explained everything as to why it would happen. But if we take into account that this happened a long time ago. What happened about in between 700 and 800 AD? What was going on in Europe? Well, the Vikings dominated that period. They were a loose association of pagan clans coming from what we now call Denmark, Sweden, and Norway. But they were not countries in those days. They were all a mixture of it. They're often called Norsemen, and they came mostly from the southern Scandinavian coast. They were superb craftsmen in all sorts of ways. They made the most amazing jewelry. They carved amazing things. And they were expert sailors and navigators. Of course, we've been given a different story about the Vikings now. They are made to look fierce, although I think they were fierce. They were taller than normal. They were certainly taller than the people that they fought against. And we see them now with these horns. But there's no evidence that the Vikings wore horns. So, but they certainly had helmets, and they certainly were fierce fighters. And they traveled in ships, and they traveled in the most remarkable ships imaginable.
In the 8th century, the world faced a new naval power. The Vikings, who set out from Scandinavia in search of adventure, conquests, and knowledge. Envied as explorers and feared as invaders, the Vikings stretched their reach to the edge of the known world and beyond. They built an empire on the back of a key piece of technology: the sleek, agile longship. The longships employed an innovative clinker-built hull, where the planks overlap each other. The design was flexible, required less water displacement, and was extremely stable, thanks to its unique keel configuration.
You have the keel to support when you're sailing, but this here have an extra keel who is built in the hull. You start with the keel, you continue with 6 planks, which is going up like to make the bottom, and the 7th board stay vertical.
And that's this one here?
That's one, yeah. So therefore, when you sail, you get 1 extra keel who support you for the direction. After that, it's continue, flat again, and go to the upper strike.
So it's sort of like a triple keel?
Yes, triple keel. More stable for sailing.
With the longships, the Vikings were able to cover vast distances quickly, mastering both open seas and shallow rivers. They sailed to Britain and France, to America, through the rivers of Russia and Ukraine to Istanbul, and even to distant Egypt. Elements of the impressive longships were adopted wherever the Vikings sailed. You can still see the influences today in Portugal's Rabeio boats and the gondolas of Venice. To give me a better sense of what it took to actually build a longship, Geir walked me through some of the early stages and explained the importance of choosing just the right tree.
When we try to split it, sometimes if the trunk is really good, then we can split this for a half hour in 2 pieces. But in worst case, it can take 3 days.
An expert like Geir can read the flow of the tree, which is extremely important to maximize the yield.
When we split this timber, we split it in the marrow lines. We first split in 2 pieces like this, and so in 4, so in 8, and so in 16 pieces.
The process of extracting those planks is really straightforward, but it isn't easy.
Now you shall get the honor.
Good.
Yes,
Hey, I'm getting the hang of this.
Yes, you're strong also.
Yeah, I'm a Viking.
Slowly, just as Geir predicted, the tree began to open.
You can see the little crack, and I follow this marrow line here now. Now you can see it's follow the marrow line down to the center of the tree.
Then it's time to turn the sections into planks. Another extremely labor-intensive task that required every available Viking. This large plank is then marked and split into smaller boards that are lashed to the ship's skeleton with whale baleen, a part of the whale's mouth used to strain krill from the sea. It made a strong yet flexible binder.
They make it to these strips, and then they binding it between this two hole here,
Yeah.
two hole here
Yes,
in the
right.
frame,
Yeah.
and they just sew it like this.
To prevent leaking, a coat of thick tar and wool rope were placed between each side plank. These planks are also riveted together using iron nails.
When you do it this way, you can see then you get flexible construction, and you can see this here is possible to move a little.
Yeah.
It don't fight against the waves. They cooperate with them and just fall a little bit off.
The world's fascination with the Viking longships has never waned. They've been studied endlessly at locations throughout Scandinavia and the rest of the world. Most famous is the Oseberg, on display at Oslo's Viking Ship Museum. A vessel so treasured, the new Oseberg Ship Foundation spent years building an exact replica to breathe new life into this ancient form. In 2012, this legendary ship was reborn. Known as Saga Oseberg, every inch, curve and angle of the original was faithfully reproduced. Today she's a window into the Viking Age and the technology that literally reshaped the world. The iconic Viking longships remain one of the most recognized and admired ship designs ever created. A vivid symbol of exploration and discovery.
Well, what did they do? Well, the first well-known raid happened at a monastery in northern England, which was on a little island and very suitable for landing shallow draft vessels. And it set the pattern for future raids. And the whole sagas were recorded by the monks themselves, who, of course, came off second best. And they recorded them as ruthless hordes of pagans who murdered the monks and stole their treasure and their women. And here is a depiction of one of these raids with the Vikings making off with all sorts of goodies and killing a poor old priest here. If we look at these events that happened in this period from 1700 to 700 to 1066, we see a number of important events. The first was the Lindisfarne raid, which was the first well-known raid of a monastery that happened again with the Vikings. But there were many, many others. As you can see here, from 793 until 830, there were many raids on Scotland, Ireland, and England, and particularly Ireland, where you can see the red dots and the orange dots indicating where they are known to have raided monasteries and stolen goods. But they were also raiding France, and as you can see that these also happened. The red dots are the early ones, the yellow and orange dots are later ones. So at a later stage, they started raiding France as well, and more about that later. But from about 830 onwards, the Vikings began to settle, and they started to settle alongside the people that they had invaded. And then in 911, Rollo founded Normandy, that area around here. And in fact, in conflicts with the French, set up Normandy. And then in 1066, one of his ancestors invaded Britain as William the Conqueror. But the important thing to realize about these Norsemen was that they were extremely successful, mostly because of complete integration with the population that they invaded. They married them, and they converted eventually to Christianity, which gave them much greater cred. And in fact, so much so that Britain was heavily dominated by the Danes. And in 884, this was the picture. This whole area here was under what they call the Dane law, which was the name given to the Danish dominance, and had quite a lot related to the fact that if you wanted to keep in with them, you had to pay them. And so they paid them what were called Danegeld. And then, of course, a well-known king, King Canute, reigned from 1014 to 1035. And he, in fact, not only reigned over England, but also Denmark, a bit of Sweden, and Norway. And these were his domains, which takes us up almost to 1066. They had such an influence on us that many of our words actually have Viking influence. And there are a whole lot of nouns and adjectives and verbs, which you can see here, which are thought to have originated from Scandinavian languages. But it wasn't only language they took around, they also carted around this mutation, this 282Y mutation. They did the same in France, where it was the rivers that they went to particularly. They sailed up and down the Seine, and they were able to get to Paris. And in fact, in 885, they set siege to Paris, but they didn't succeed. And eventually they formed a treaty which allowed them to take over this large area, which is Normandy. But other areas were equally affected. Down in the south, similarly, going along the Loire River, and then also in Rouen itself. And Rouen was particularly important because it was there that Rollo set up his base. And there he formed Normandy with the agreement of the French kings. And as he was partly related to English royalty, he claimed in 1066, he claimed the throne by landing at Pevensey, and at the Battle of Hastings was able to kill Harold and so take over England. We've seen these boats before, and this is the Bay Tapestry showing the Normans crossing the sea to invade England. And here is a depiction of Harold, who got an arrow in the eye, and so brought the battle to an end. Poor Harold had spent the whole night up traveling from another battle that he'd been fighting the day before. Wasn't in good shape, I don't think. If we now look at how frequently we find the C282Y chromosome, we find, as you see in red here, there's this area here, which is very much the heart of hemochromatosis. And it fits exactly with the area that was occupied by the Vikings. And it gets less and less and less as you travel eastwards. Bulgaria hasn't got any, because of the less and less influence. The numbers were much higher here, therefore the transmission of this gene was higher in this region. And so it's in this region that we find hemochromatosis most commonly. And of course, C282Y has colonized the earth, be it in South Africa, Australia, Canada, New Zealand, the USA, everywhere that Western Europeans have gone, the 282Y gene follows. Now, why should a gene like this be so powerful? Well, something is selecting it. Well, first of all, if you carry the gene, you are less likely to get iron deficiency. And iron deficiency, we know, is a potent cause of fetal loss, maternal mortality, and interestingly, you get improved temperature regulation and you survive better in a cold climate. Well, what could make you survive better in England than having the C282Y mutation? Also, there's low, because the macrophage doesn't have iron in it, it changes the way the macrophage works, and it makes it much more efficient in coping with some diseases, particularly tuberculosis, but there are a whole string of other diseases that it helps along with. And it's also associated with a decreased risk of Alzheimer's, Parkinson's, and amyotrophic lateral sclerosis, decreased atherosclerosis, which, as we know, leads to heart attacks and strokes, increased life expectancy, increased height, which was of great interest because the Vikings were noted to be tall people. And it also, in its heterozygous form, increases the activity of sex hormones. In its homozygous form, it decreases it, but in its heterozygous form, it increases sex hormones, giving these people a reproductive advantage. Another thing I like really is that a recent study has shown that 80% of the 2017 Olympic athlete team carry the gene, suggesting that they've got power that the others don't. And compare that to 27% in the French population. Well, now, as we've mentioned, hemochromatosis is a progressive disease, but there's a time when to start therapy. Many of the patients we see them right at the end of the thing here, and starting therapy there, although it helps and prolong life, it doesn't take away the problem. But if we start about here, we can take away the problem. And the way to do it is simply to take blood. We do it a bit more in a much more sophisticated way than then, but nevertheless. But interestingly, in a severe case, it's done once a week. Now that seems an awfully long time, and it can be done once a week, provided the person has an adequate nutrition and has got enough folic acid. They can make blood fast enough for you to take blood every week. Once the iron has become depleted, it has to go on about between 2 and 4 times a year. And the response is an improvement in fatigue. Cardiac function can completely return to normal. Liver fibrosis, early fibrosis may diminish, but if cirrhosis is present, it doesn't go away. The skin pigmentation improves and the bone changes improve. But the arthritis doesn't get any better. The diabetes doesn't get any better. The pituitary doesn't get better. And most important, the cirrhosis doesn't get better. And 30% of the people who have cirrhosis will go on to develop liver cancer. So let's see what happens when we take it. We go back to our old picture again. This is where we were before. I've put in a nice picture of a cirrhotic liver to give you an idea about what's going on. But the first thing that we do when we take blood is that we take red bloods out. So we bleed them weekly. This causes a massive increase in the amount of iron, amount of red cells that are needed and are made by the bone marrow. And of course, that increases the amount of iron that they need. And that iron comes here from the liver. As this progresses, so you need less and less going to the macrophage. And so less and less iron comes from the macrophage. And so you end up with a simplified circuit where most of the iron is coming from the transferrin and going into the red blood cells, which is then being lost into the bottle and hopefully into a donor at the other end. The amount of iron that comes in here is really not affected. And many people have thought, oh, well, we could treat hemochromatosis by just limiting the amount of iron that you absorb. And that just doesn't make sense because you would have to start many years before they were born in order to get the numbers right. But however, it does make sense to reduce high iron food levels in patients with hemochromatosis, although it's not going to cure them. This is what happens. Here is an example of serum ferritin getting lower and lower as you, that's meant to be weekly, sorry, weekly venesections. And when you get to a point where you deplete the level of iron, round about when the ferritin's there, you get to a point where suddenly the transferrin saturation, which has been high all the way along, suddenly drops down. And if you go too far, you get into iron deficiency anemia, where the person would become anemic. So obviously that's why we can't go on giving iron taking iron in the form of blood every week. So reduce the tempo down to about 2 to 4 times a year. Well, is it possible to use iron chelation? Yes, it is. More recently, we've found very good iron chelators, but they're expensive and it's not as altruistic. The blood that you take from hemochromatosis can be used for other people. And if you just use iron chelation, you can't do it. But there are people where venesection is impossible. And there are some situations where you need to speed up and go faster than 1 a week. So for instance, in severe cardiac failure. And so you can add a chelating therapy on top of the venesection, which will speed the process up. But if you relied just on the chelation, you would get the same effect, albeit somewhat slower. What happens to these people? Well, non-cirrhotic hemochromatotics, like us all, they die off. But cirrhotics die very much faster. And as I said before, mostly because about 30% of them will develop liver cancer. Hagar always has the last word. He's been a Viking all his life, got nothing much to show for it, but he has some satisfaction. In fact, he knows that he'll live longer, that he's more sexy, and that his descendants will dominate the world. Thank you. Well, I'm happy to take questions.
Can I ask you one, Patrick? Gee, that was fantastic. I mean, it's no longer a mystery why you're a professor and I'm not. But what I wanted to ask you was, is there any place for diet in hemochromatosis? Obviously, if you had less meat, that might help. But what about broccoli? Was George Bush right in hating broccoli?
Yeah, well, that brings up a whole another area. Iron comes in 2 forms in our diet. One is in meat, and obviously, if you drank blood, you would get a lot of iron as well. And that's obviously you can cut out meat altogether. There are lots of people who live a vegan life without too much trouble. When it comes to non-heme iron, the iron in vegetables and things, it becomes a much more tricky situation, because most of the iron in vegetables is not available to us. So, for example, the myth of Popeye getting his strength from eating spinach is rubbish, because spinach contains so many inhibitors of iron absorption, mostly polyphenols, that it doesn't work. In fact, you absorb very little iron from spinach, and similarly from broccoli for the same reason, although it's got lots of iron in it. However, if you take it with something that enhances iron absorption, you can actually get a good picture. And so if you take it with things that have got vitamin C in it, particularly, vitamin C is a potent enhancer of iron absorption. So if you have hemochromatosis, you should avoid too much vitamin C because that will enhance iron absorption. It does nothing to heme iron absorption. It only works with non-heme iron absorption. It's an area that's difficult. There are pressure groups that have tried to pressurize hemochromatotics into taking on a completely iron-free diet. And the problem is that it's not palatable in many respects, and it just doesn't work. Although it will help a little, it will never help enough. And so mostly you find people with hemochromatosis cut their meat down if they can, and rely on being venesected.
Interesting. Thanks, Patrick. Very, very good talk.
Patrick, if I can ask, the thing that fascinates me about this presentation and some other presentations that you and other people have given are these complex circuits in the body that has many chemicals and many proteins and many processes. How long has it taken to unravel this particular complex circuit that you've been talking about?
Well, I think it's fascinating because, in fact, it hasn't taken all that long. I think that the first inkling that iron moved around the body, where in a cohesive way, was in the 1930s, where people began to work out these things. But you know, the role players were not known, and also the idea might have been obvious that, you know, iron gets into the body and it gets through somehow or other, but nobody really knew the steps. And it's been a gradual thing. I write a chapter in a book, and I've just had to update it. And I haven't been really at the forefront of what's going on in iron, but what's happened in the last 10 years is frightening, because there have been massive increases in the amount of what we understand about each of these proteins and about how they work, and in fact, their very existence, things that we didn't even know about, have emerged in the last 10 years. Not major role players, but nevertheless things that do explain a little bit. So they've evolved, and I think that they've evolved over a period of about 150 years, I would guess, because I think before that there was no knowledge.
And when was the treatment to blood victim? When was that discovered? And since when have they done that?
Since 1930, interestingly enough, long before they really understood how it was working. In 1930, somebody proposed that we take blood. You know, after the First World War was a watershed in blood transfusion, because it was only during towards the end of the First World War that people realized that they could take blood from one person and give it to another. It had been done before, but with all these remarkably haphazard results. Sometimes they gave blood and the person got better. Sometimes they gave blood and they killed him. And they never knew why, and they didn't understand why this was until we really began to understand what blood groups were. And so when blood groups were first invented, transfusion became possible and donation became more likely. Of course, in the early days, they used to bring the patient to the bedside and fasten up the patient to the donor and just pump the blood directly across. But nowadays, there's a whole sophisticated way of doing it. And as that has evolved, people began to look at venesection. It's interesting that although it started in the 1930s, it didn't really catch on until later. And in fact, in my early career, I spent many, many hours fighting with the blood banks, getting them to use the blood, because they were unwilling to use it because it seemed to be somehow contaminated and may do their patients harm, which was nonsense. And in fact, it's now most blood banks, or in fact, all blood banks, I would think, utilize the blood freely. There are problems with HIV and things like that, because somebody who's being bled every week, it means there's a supply coming every week. And what he does in between while, as he gets better from his hemochromatosis, he might become adventurous and develop AIDS or something, or get to the HIV infection. And that has to be detected. So obviously the blood goes through all the screening processes, even though it is collected every week.
In the days when they used to bleed people almost all the time as a sort of ignorant way of trying to address a multiple range of diseases with their niches, they were probably doing some people good without knowing why.
Yes, I'm sure that that's so. But I think they mostly did people harm. You know, the whole story of anemia, it's a fascinating story going out through the ages, because it used to have the name chlorosis, and it gave people, when you become quite severely anemic, you get a sort of faintly green color, and that was the origin of the name chlorosis. And it was associated with beauty, and in fact, if you go into the art, of medieval art, you'll see quite a lot of women who have this slightly green color to their skin, which was thought to show virginity and the like, purity. Iron deficiency anemia, which is still a major problem today, in the Middle Ages and before was a very big problem. And so I think that bleeding such a person probably hastened their demise.
Now, you've touched on the opposite problem. If somebody is anemic, does it mean some of the systems you've been talking about are deficient in a different way?
Ah, yes, those are fascinating. They are very rare, surprisingly. Iron deficiency is the antithesis of iron overload. And as the only way you can get iron in is through your diet, it's not surprising that the diet is an important part of iron deficiency. And in fact, iron deficiency in the world today is most commonly due to a combination of poor iron nutrition and blood loss, on the other hand. So that explains why in places like India, large numbers of people are iron deficient. It's because the diet that they have is very deficient in iron. Not so much that there isn't any iron in the diet, but there are a large number of inhibitors that prevent the iron being absorbed. And so that would be a diet rich in rice and rich in vegetables and so on. But of course, bleeding does play an important role in iron deficiency. And it used to be said that you must make sure that the person's not bleeding first. But I think more recent work has shown that whole populations can be iron deficient without bleeding to any severe extent. So it obviously makes sense that women who menstruate regularly, for example, or who have multiple pregnancies are likely to become iron deficient because they're losing blood and therefore losing iron, and they have to pass a good amount of iron onto the fetus. In fact, much more than the iron that they would have lost through those 9 months of missed menstruation. So pregnancy and menstruation play a big role in iron deficiency in women. But the primary cause is failure to take it in a reasonable diet. If you look at the different diets, if you look at different countries, compared Western diets to non-Western diets, you'll see that a lot more iron is available. About 15% of the iron available in a Western diet. That's a diet with a lot of vitamin C in it, a diet with a lot of iron, probably with meat in it and so on. About 15% of the iron is available. Compare that to a diet at the other end of the scale where it's mostly pulses and cereals, and about 5% of the iron is available. So it's a factor of 3 between the two. So that's the other end of the story. There is an interesting and new and modern twist on iron deficiency, and that is that we are beginning to find genes that code for some of these iron proteins that don't function properly. And instead of causing iron overload, they cause iron deficiency because the iron is not being absorbed properly. And this is becoming an interesting field, although the individuals who are involved are very, very rare. So it's not a widespread problem at all.
Patrick, thank you very much. This has been a very, very interesting presentation. Thank you for all the preparation, for all those beautiful slides. It really was both from a historical point of view and a medical point of view, a very interesting presentation. So thank you for all the work you've done.